So this is what we were looking at. Okay, so this is the absorption spectrum, and this is for chlorophyll. It's different for other pigments. Remember, plants have more pigments than chlorophyll. Right now, you can see outside that the leaves are changing and they're losing their chlorophyll, and that's because the sun's intensity is not as strong, and so they've already stored up a lot of their starches, etc., for the fall. So they're losing their chlorophyll right now. But the chlorophyll is the main photosynthetic pigment. It absorbs the energy from light and makes the light reactions possible. So if you look at this, plants have chlorophyll B and chlorophyll A. What are the main colors that they absorb in? Blue and red. And then if you look, they absorb almost no green, and that's why chlorophyll is green. Remember, the color that you see is the color being reflected. So it absorbs blue and red, and it reflects green. And there are some pigments that can absorb at different colors, but chlorophyll is the main pigment, and it absorbs at red and green. You don't have to memorize the wavelengths, the 400, 500, 600, 700. I think you guys did that probably in middle school. You did the different spectrums. I didn't mean to change that. It'll usually be listed for you, but like 400 is blue, and then red is in the 700s. We might at some point, if we get time, do this. We can separate pigments in the plant into its different parts. So you guys have this. Now we're on that packet that I gave you on Friday. Okay, not the first page. This is the second page. Okay, but to save time, these first few questions are really easy. So I want to start on the light-dependent reactions page. I think I skipped a page. Nope. That was it. So 1 through 7. Okay, 1 through 7. I'm going to let you guys answer on your own as a review. And I want to look at this model 2 with you. This is a picture of the inside of a thylakoid. This is the light-dependent reaction. So it would be more of a coin shape. Just because a lot of times we have trouble visualizing what this actually looks like in the cell. Yeah, so this would be one thylakoid in a whole stack. Okay, you can see, there we go, there's my pins, that it has a membrane that looks a lot like the cell membrane. That's because all of these organelles that have their own membranes, remember, it's made out of phospholipids. So it has phospholipids that are separating I don't know if it's going to let me mark yet, the inner thylakoid. Separates the inner thylakoid from the stroma. So remember, the light-dependent reactions happen inside the thylakoid, and the stroma is where the Calvin cycle happens. You're about to find out why the light-dependent reactions absolutely have to happen in the thylakoid, and that's because of all these H+, these hydrogens, these protons are required to run ATP synthase. Okay, so this is one of those things where there's a lot of things happening at once, and I am going to try to point them out to you one at a time, and then we'll kind of try to put it together. So I'm going to start at photosystem 2 as we start to talk about this, but this is really all happening at once. So the first thing to see is on one side of your membrane, you have a photosystem 2, a photosystem 1, and sitting between the two is an electron transport chain, which does exactly what it sounds like. It carries that electron through the membrane to the next photosystem. One thing that's confusing about this is that they're named out of order. Okay, so when you're looking in the membrane, photosystem 2 is where our models start. Okay, so it goes 2, 1, sitting in the membrane. Okay, the reason it's like that is because they discovered this photosystem here. Photosystem 1, they discovered first. So it's named by the way it's discovered, not the way it's in the membrane. So think about that first. Let's review really quickly because it's important to understanding this. Oxidation and reduction reactions. If I'm using oxidation, if I say oxidize or reduce, you guys need to know what that means right off the top of your head. So, do you guys remember learning, I don't know if you learned this or not, oil rig? Okay, oxidation is losing electrons. Does that sound vaguely familiar? No? That's pretty ugly. But oxidation is losing electrons. And this is what's confusing about it. Reduction is gaining electrons. And it's kind of like, whoa, whoa, whoa. Why is it called reduction if you're gaining something? What is the charge of an electron? Is it negative, positive, or neutral? Okay, it's negative. So what's being reduced is the charge. When you gain an electron, like when a molecule gains an electron, it reduces the charge because the electron is negative. So that's what you have to remember. I used to always get this mixed up in school because I'm like, reducing, that's losing. But reducing, what you're reducing is the charge. You gain a negative electron, you reduce your charge. Do you guys remember what part of a molecule the electrons participate in? You've got the protons and neutrons in the nucleus, and then the electrons are parts of the bond, right? They're the bond that's holding the molecule together. So you can, in chemistry, it's not really taught to you this way, but I'm trying to make it the way you can understand in here. Electrons are like energy. Okay, when you pass off electrons, you're passing off potential energy to the next electron carrier. So that's what you have to remember. Electrons are energy. When electrons are charged and they're high energy, that energy can be passed on to the electron character, the electron acceptor. So we've got oxidation and reduction happening this, okay? What happens? Remember, what is our energy source for this reaction? Not yet. That's going to be the energy source for Kelvin. What powers this process? Light. Let's label it. Okay. Light is the energy source for this reaction. So light strikes the photosystem. Okay. And look at what's sitting at the top of the photosystem. What is this? This is the light dependent reaction. We're not in the Kelvin yet. Kelvin's second. First, we got to charge our batteries. So that's basically this, but like in depth. Yes. Yeah. Okay, that's what I'm saying. Today we're doing light dependent, and Wednesday we'll do Kelvin cycle. Kelvin cycle won't take as long, so we'll do some other things that day as well. Okay, but the light strikes the chlorophyll. It's attracted to the chlorophyll. Okay, chlorophyll is embedded in this protein. Sitting on this protein is a high energy electron. That's what the black is. So maybe highlight that to point it out to yourself. The black dots are high energy electrons. Okay, so in every photosystem, there is an electron. Okay, the photon hits it, excites the electron, transfers the energy to that electron, and it starts traveling through the electron transport chain. Okay, this is one part of it. When it arrives here, okay, look, it's going to be, there was also an electron resting here, and it's going to go and be a part of what? What does this electron join with? NADP plus. Okay, NADP plus. Here's where your hydrogens come from. This is what's confusing. Electrons are negatively charged, right? Very negatively charged. So when an electron is traveling through the electron transport, or over here when it's being donated to NADP plus, because it's so negative, it's going to attract hydrogen in the cell. Why would a cell have an abundance of hydrogen? What are cells majority made out of? Living organisms are 50 to 70% water, okay? So because there's water in the cell and the hydrogen of that water is participating in these loose hydrogen bonds, hydrogen protons are very readily available within the cell, okay? And they're really easy to move for a lot of different reasons that we really don't get into in here. So when this electron starts moving through the electron transport chain, it's attracting hydrogen and pushing hydrogen into the center of the thylakoid. Do you guys see that? Okay, so two things are happening with this electron so far. Okay, this electron has become excited and it's moving through the electron transport chain. As it's moving through the electron transport chain, it's pulling hydrogens, it's attracting hydrogens through those proteins into the center of the thylakoid. Okay, when it comes over here, okay, it is replacing an electron. Okay, the electron that was already here becomes excited and joins NADP+. So when this NADP+ receives the hydrogen that's being shown here, it's also receiving electrons. Okay, so NADP+ is not charged. Okay, it receives that electron, and because that electron is so negative, it also attracts a hydrogen, and that's how you get NADPH. It's the electron plus the hydrogen that it's carrying. Okay, so when it makes that new bond, now there's a high energy bond that can be used when we get to the Calvin cycle. Okay, so that's one part. All right, so far, and then I'll explain. I'm gonna repeat myself, and it's gonna be annoying, but it's because you could start at multiple places here. All right, well, when this electron, there was an electron sitting here, right? And it left, and it went through the electron transport chain. Well, this photosystem is useless until it has another electron. Okay, it has to have another electron to participate in this reaction. So water is where that electron is coming from. Okay, light strikes the photosystem, excites that electron, and also splits water. Okay, and when you split water, look, do you see this little guy right here? Okay, when you split water, you harvest the electrons from water, and you also release what two things? It's H2O. So you're releasing oxygen and hydrogen. Okay, so that is also contributing to this gradient. Okay, so light strikes the photosystem. It excites the electron, and this electron moves through the electron transport chain. In the process, it pulls hydrogen into what we call the lumen, or the center of the thylakoid. Okay? Also happening when light strikes, water is split, releasing oxygen and hydrogen, contributing to this high concentration of hydrogen, but also providing, that's right here, a new electron for this photosystem. Okay? Meanwhile, over here, there was also an electron, right? Light struck this photosystem, excited this electron, and it becomes part of NADP+. Okay, so this is one of your energy carriers. NADPH is one of your energy carriers that is made from the electron at photosystem 1. Okay? But now that this electron is gone on photosystem 1, remember, it's useless until it gets another electron. So where does photosystem 1 get its other electron from? Photosystem 2. Okay, so the electron on photosystem 1, which is right here, is being replaced by the electron that came from photosystem 2. Okay, what replaces the electron that left photosystem 2? The electron from water. Okay, so that right there is like half of it. What replaces the electrons? Okay, at photosystem 2, the electrons harvested from water, because when I break a bond, I get electrons, I release electrons, okay? So this one, photosystem 2's electron, is replaced by the splitting of water. Photosystem 1's electron is replaced by the one that traveled from photosystem 2. So is it like you're in a factory, like a moving belt? Yes, it's like a moving belt, and if one part stops, the other part can't function. I'm confused about the whole Okay, the reason, my only gripe with this diagram is it's showing it balanced. Okay, this has to happen twice to get NADPH. So that's why it's showing you 2. Because it's a flat diagram, so it can't show you 2 rounds. But it takes 2 rounds to make 2 NADPH. The electrons, okay, remember electrons attract hydrogen, right? So the NADP plus accepts the electrons and hydrogen, and that's how you get NADPH. Because what you got to remember with this is anytime you have an electron traveling along in the cell, hydrogen is probably traveling with it because that negative electron attracts the hydrogen.
So you already have NADP plus and
You, the electron is what allows it to bond together, right? Okay. Now, what energy carrier have I not addressed? I haven't addressed ATP. It's being made down here at ATP synthase. Okay, ATP synthase is a protein, okay, that uses an electrochemical gradient to make ATP. All right, look here. Through the electron transport chain pulling in hydrogens and through the splitting of water, I have built up a higher concentration where? Inside the thylakoid or outside the thylakoid? Inside. And they're going to want to move out, right? Because molecules move from high to low. Okay, these positive hydrogens are going to move through ATP synthase. And they're going to move through with force, kind of like, have you guys ever seen a water wheel, like at a mill, how the water causes the wheel to move and then that energy is used to run the mill? That's kind of the analogy I would use for this, because this hydrogen moving through causes the ATP synthase to actually spin, okay, and creates like a kinetic energy that allows the synthesis of ATP. Because if you'll remember from when we talked about ATP freshman year, ATP, adding that third phosphate to ATP, okay, it was ADP, right? When I add a phosphate to it, creating that bond stores energy. Okay, so it takes energy to put that molecule together. And the energy is coming from the movement of ATP synthase, from the spinning and the movement of ATP synthase. ATP synthase is powered by the proton gradient. It's powered by the proton gradient. And where did the proton gradient come from? 2 places. Yeah, the photosystem 1 where I split water, there was some hydrogen there, and also through the electron transport chain. As an electron travels through the electron transport chain, it pulls hydrogen into the cell. Because that electron is so negative, it attracts hydrogen. Okay? Are we kind of there? So you have a bunch of stuff happening at once, okay? You've got the splitting of water, which creates oxygen and gives up an electron. Okay, you've got an electron moving through the electron transport chain and pulling hydrogen in. The electron that traveled through the electron transport chain replaces the electron at photosystem 1, I about called it wrong, photosystem 1, and that electron becomes part of NADP plus, charging your other energy carrier, NADPH. Okay, so you've got 2. This is called oxidative phosphorylation. This process with ATP synthase is oxidative phosphorylation. Okay, because what is driving it is the electrons moving, right? Was water oxidized or reduced? Look at what you wrote on the top of your page. Is water being oxidized or reduced? Oxidized. Because it's losing electrons, right? So when it loses its electrons, it's also causing hydrogen to build up from the splitting of water here and from the electron transport chain. So because you're oxidizing water, you're building a proton gradient that allows you to power ATP synthase. So that's why ATP synthase is called oxidative phosphorylation. Phosphorylation, adding a phosphate. You're able to add that phosphate because you built a proton gradient when you oxidized water. Okay, what is being oxidized in this? Water is being oxidized. What is being reduced? Who is accepting an electron in this? NADP plus, okay, is being reduced. So these are oxidation and reduction reactions. If I puncture this membrane, if I pop a hole in this membrane, can this process continue? No, because my hydrogen is going to leak out. And then I don't have a gradient, so I can't run ATP synthase. Okay, so what do you need to know about this? You need to know the purpose. What is the purpose of the light-dependent reaction? To make your energy carriers ATP and NADPH. Okay, so my purpose is to make electron carriers. What is the energy that's powering this process? Sunlight. Okay, the sunlight is powering the process. Where does the electron come from that replaces the electron at photosystem 2? Water. Where does the electron come from that replaces the electron at photosystem 1? Good, photosystem 2. What's powering ATP synthase? The hydrogen, the proton gradient. Okay, what's the special name for that? Oxidative phosphorylation. And you're going to have it in respiration as well. Anytime you have ATP synthase, that is oxidative phosphorylation. You can also have something called substrate level phosphorylation that's made when the electron from one molecule is given up to ATP, and you're going to see that in respiration. Okay, but right now the ATP synthase, the oxidative phosphorylation is what we need. And it's also important to know, why does this have to happen inside the thylakoid, inside a membrane-bound structure? What can you not build without a membrane-bound structure? You can't build a gradient. Okay, that's why it has to happen in the thylakoid. If you lose that membrane, if you rupture that membrane, you can't make ATP anymore because you don't have a proton gradient. Okay, so in Model 2, what shape represents the single electron? The black dot. Mine's green, but yours is black. Okay, 2 places where electrons are released by a photon of light. That is your photosystems. So this is PS, let's write them in order. PS 2 and 1. Remember, they are backwards because 1 was discovered first. And when they discovered 1, they knew there had to be something else. Because they were like, this electron has to be replaced by something. This electron has to come from somewhere. So that's one of those cases in science where they kind of made a prediction that there would be a second photosystem before they actually found it. Find one place where electrons are released from water. That's photosystem 2. Okay, when the electrons are released, what other products are formed? Hydrogen and what else? And oxygen.
Wait, where does the oxygen go in?
Out of the cell. That's a good question. Eventually, it makes its way out of the cell. Remember, do you guys remember that CO2 and oxygen can readily diffuse across membranes? Because it's so small, so it just diffuses right out of the chloroplast eventually, and then it's released from the cell. Yeah, so after this happens for a while, and that's a really good question. After this happens for a while, oxygen builds up, and then it just diffuses high to low out of the thylakoid and then out of the chloroplast. Okay, next, these are the rest of the questions for this. Write A, B, and C to match this up. Excited electrons leave the chlorophyll and reduce NAD plus to NADPH. That's A. So where should I put A? What photosystem does that happen at? Okay, that's happening at photosystem 1. So label that with an A. And guys, everything that we've talked through right now is also written out in the PowerPoint, but I just much prefer this diagram, and that's why we're looking at it here. Excited electrons move through the electron transport chain, providing the free energy needed to pump hydrogen into the thylakoid. Okay, that's going to be B. It's not quite happening at 2. It's happening in between. It's happening at the electron transport chain. And then that means that this is C, okay? C is hydrogen, oh, nope, just kidding. Hydrogen ions flow out of the thylakoid via the protein. That's not here. I got ahead of myself. That's where. Where do they flow out? Okay, hydrogen ions flow out, providing the free energy needed to convert ADP to ATP. Okay? All right, on 11. Okay, we've got 4 minutes, so I'm thinking maybe we're just going to do this together so that you have it. Briefly describe how the system works and what job it performs. What job does the electron transport chain do in the membrane? It moves an electron, but what else does it do? Yes, as electrons move through, because this is the most important part. That's why you have a whole question about it. This is the hardest to understand and most important part of the light-dependent reactions. As electrons move through the ETC, hydrogen is pulled into the thylakoid, building a what? Yeah, building a proton gradient. Okay, and make sure that you know H plus and proton interchangeably, because you could see both of them. It could say a hydrogen gradient or it could say a proton gradient. Okay, and then which protein in the thylakoid membrane uses excited electrons to reduce NAD plus to NADPH? That's photosystem what? It's the second photosystem, but it was found first, so it's photosystem 1. Okay, which provides excited electrons to the electron transport chain? Photosystem 2. So this POGL literally has everything that you need to know in these 2 pages that we're looking at, okay? That's why we're doing it together. And then the embedded protein that converts ADP into ATP using the energy from the hydrogens. What is that? ATP synthase. Okay, once the chlorophyll has been released, it's no longer useful until those electrons are replaced. So what replaces the electrons at photosystem 1? You guys can look at your front page if you want. Yeah, the electron from photosystem 2 that traveled through that electron transport chain. Okay, what replaces the electrons at photosystem 2? An electron from what? Yeah. Is carbon dioxide involved? Okay, no, this is like the biggest thing I battle with. No carbon dioxide. Is any glucose being made? No, this is like your warm-up. You're charging your battery so that you can actually do that. We're going to skip A for a second because we already did it. All right, all of this is math that you guys don't have to calculate anymore. Okay, so we're not going to do this right now. If you want to cross it out, okay, because all the important stuff is right there. And what I want to do now is I want to give you guys an FRQ to at least start. And the FRQ that they did in class, you ladies will need to make up when you return to school. We are not doing FRQs at home anymore. We can discuss why if you have any questions, but I will need you guys to make time to make up those FRQs when you return to school. You're welcome to do it during your AP prep period.